The light reactions typically occur in the way that we've been talking about in the previous video. This is also called non-cyclic electron flow. So again, this is usually what's happening and what we've already been talking about. It involves PS2, PS1, ATP synthase, and NADP plus reductase. It's a linear process, so non-cyclic also means linear. So there's a start and an end. It produces equal amounts of ATP and NADPH. So again, it's what we've already been talking about. Over in PS2, energy is absorbed. That energy is sent to the reaction center where an electron is boosted and then passed to an electron transport chain. The electron transport chain harvests the energy to move hydrogen ions against their gradient. That gradient is used to make ATP, and then that electron gets passed to PS1 where light energy also boosts the electron and then passes that energy-rich electron to make NADPH. So I could also write this out as electron flow. So where does the electron start and where does the electron end? So if you think about PS2, the ultimate source of electrons is water, because water is going to lose that electron and never get turned back to its normal state. Everything else will gain an electron and then give it away, gain an electron, then give it away. So everything else just cycles between having an electron and getting rid of that electron, but water actually is permanently changed by giving away its electron. So really, the electron starts in water. That electron is then given to PS2, and that's the P680 inside of PS2, because it has to fill that electron hole in P680. It's then passed to a primary electron acceptor. The primary electron acceptor passes it to the electron transport chain. It goes down the electron transport chain, and then the electron eventually lands in PS1. And so remember, that's P700 inside of PS1, so filling the electron hole in P700. PS1 then passes it to NADP plus reductase, which puts that electron into NADPH. So that electron is going to end finally in NADPH, and then that will, of course, eventually go to the Calvin cycle, which is a topic of later video. Okay, but for non-cyclic electron flow, again, it's a linear process. The electron starts in water, gets passed to PS2, passed to a primary electron acceptor, passed the electron transport chain, passed through PS1. Oh, I forgot my primary electron acceptor here, so adding one step here. It gets past the primary electron acceptor and then finally ends in NADPH. So again, non-cyclic, it produces ATP and NADPH and oxygen. It uses PS2 and PS1. It makes equal amounts of ATP and NADPH. So for every ATP, one NADPH is also made. So that's great, but there's a problem. Oh, hold on, I'm getting ahead of myself. So this is just another figure about non-cyclic electron flow. I like this figure. I put it on D2L just because I think it's really simple and straightforward. So here's the light harvesting complexes. They absorb the light energy. They pass that energy to P680. The electron is excited and taken away by the primary electron acceptor. Water puts its electrons into P680, and then the electron gets passed down into the electron transport chain. The energy from that is used to make ATP by a gradient. And then over in PS1, same thing is happening. Light is shining in, exciting the electron, and passing the electron to NADP plus to make NADPH. So again, we have equal amounts of NADPH and ATP, and we're also making oxygen using both PS2 and PS1. However, the plant will also need to do something called cyclic electron flow, and it's going to have to do this because the Calvin cycle uses more ATP than NADPH. So non-cyclic electron flow, the one we were just talking about, makes equal amounts of ATP and NADPH, but the Calvin cycle is using more ATP. So you can see that eventually the cell will run out of ATP but have plenty of NADPH. So if it runs out of ATP but still has lots of NADPH, it doesn't want to make both of them. It only wants to make the one that it needs. So because it's out of ATP only, it's going to want to carry out cyclic electron flow. Because cyclic electron flow only makes ATP. It doesn't make any more NADPH. So it's only going to use photosystem 1. The electrons are going to flow in a cyclic way. So the electrons are going to make a circle rather than a linear process. And it's only going to produce ATP, no oxygen, no NADPH. And this is what it looks like. It's using the basic equipment still. So an electron in P700 is excited by light energy, but instead of passing that electron over here to NADP plus reductase, it passes it back into the electron transport chain. The electron then flows down and lands back in P700. So the electron gets excited, passed down the electron transport chain, and lands exactly back where it came from. That's what makes it cyclic electron flow. Because it's only using the electron transport chain, it's only producing ATP. It's not using the NADP plus reductase, it's not using PS2. So again, no oxygen, no NADPH, only ATP, and it's only using PS1. And it will switch into this in order to make more ATP because it runs out of ATP first. So just to kind of wrap up the light reactions, the question is, what color is least effective in driving photosynthesis and why? So if you remember back, this is all about the ability to absorb light. Absorbed light is the light that can be used in photosynthesis. Reflected light is not absorbed and can't be used in photosynthesis. So when you look at the chloroplast, it's green, and that means it's the reflected light that's not absorbed. So green is the worst color for photosynthesis, at least in plants that are green. So what if a plant was violet? Would green be the least effective color for photosynthesis? Hopefully you realize that no, it would not be. In fact, violet would be the worst color because, again, that's the reflected color, the color that's not absorbed. In the light reactions, what's the initial electron donor and where do the electrons end up? Well, remember that non-cyclic electron flow. The electron starts in water and it's going to end in NADPH. So that's that non-cyclic electron flow. In cyclic electron flow, there's no donor and the electrons don't end up anywhere different because it's a cycle. They'll just keep going back and forth, right? So this is really true for non-cyclic electron flow.